Okay,
Okay.
how's that? That should work.
Much better. We can hear you now.
My apologies. I'm going to just talk about how we can apply evolutionary dynamics and mathematical modeling to actual cancer um, protocols. This is work that was developed at Moffitt, which about 16 years ago decided to embed mathematicians into the cancer center so that we can pretty much mimic these research paradigm of physics in which there are experimentalists linked explicitly to mathematically based theoreticians. And so we developed this kind of mixed approach to this. We now, I think, have 9 faculty mathematicians and 2 evolutionary biologists, some of which are attending the meeting, and I'm sorry I can't be there. The basic problem is this, and I don't have to, I think, sell this to anybody. This is a case of metastatic lung cancer treated with a targeted agent, lorlatinib, with a very effective response. But as the treatment continues, you get progression and treatment failure. The general principle here is that resistant phenotypes are inevitable, and that trying to interact at a specific mechanism of resistance is probably futile because there are probably many of them. On the other hand, the clinically resistant population is typically very small, and so it has to proliferate, become clinically important, and those are governed by Darwinian forces. If you look at cancer as cancer treatment as a game, and the game is being played by the oncologist who moves by applying a therapy, and the cancer plays the game by developing an adaptive response to defeat the therapy. The oncologist actually has 2 huge advantages in this game. One is that he or she plays first. It's the equivalent of white pieces in chess. In other words, the cancer cells cannot begin to adapt until the oncologist actually applies the therapy. And the second thing is that the oncologist is sentient and can play dynamically, can anticipate the future, and can alter the way therapy is administered in a way that keeps that takes into account the evolving cancer population. The cancer population, in contrast, like any evolving population, can only adapt to the here and now. It can never anticipate the future, and so that is a fundamental advantage. The problem is that that oncologists have used a treatment paradigm for the last 5 decades, and that is they apply the same treatment agents continuously at maximum tolerated dose until progression. Notice that by doing that, notice the oncologist keeps giving the same treatment over and over again, and does not change therapy until the tumor has progressed. So the oncologist loses both advantages effectively, because the oncologist does not change treatment until the tumor has progressed. The tumor is now leading the game, and the oncologist is following. So just in general, our goal here is to try to get the oncologist to play more dynamically and to engage in the game more actively. So if we restate the problem and the failure to use optimal game theoretic approaches, is let's assume that we start with a tumor that has a mixture of cells that are sensitive and cells that are resistant. We apply a very effective therapy. We have a great response. But you'll notice that the response is all the cells that were sensitive. You can continue therapy, which is the way this is done. Again, constant, continuous application of the same treatment, and there is no further benefit to the patient. In fact, by using this approach, you elicit a response to this called competitive release. In that you intensely select for resistance, and you eliminate the possible competitors to the proliferation, so they can essentially proliferate unopposed. One evolutionary strategy is called adaptive therapy. There are many others, but this is the one I'm going to focus on. Simply gives the treatment, rather than maximum tolerated dose continuously, you simply give pulses of treatment. The idea is to knock the tumor down, but explicitly retain a significant population of sensitive cells. The treatment is then withdrawn, and the tumor returns, it proliferates, but in the absence of selection for resistance, the sensitive cells usually have a fitness advantage, and as they regrow, they effectively suppress the proliferation of the resistant populations. And so you simply just do this over and over again. The idea is that by you're in this case using the treatment as a sort of a forcing function, and you're controlling the evolutionary dynamics of the tumor over time, rather than just sort of explicitly trying to kill as many cancer cells as possible. The first trial in this has been published, and I'll just briefly go through. Mathematical models were developed based on the interaction of metastatic prostate cancer cells with their main driver, which is testosterone. We defined subpopulations, and the treatment in this case was second-line therapy with abiraterone. I won't go through the details of this. We can discuss that later if you want. But the very simple therapy, just Dr. Jinzhan Zhang was the PI of this and essentially ran the trial. So the abiraterone, this is a single therapy in prostate cancer that's castrate resistant. Abiraterone is given when the PSA falls to below 50% of the pre-treatment value. Abiraterone is discontinued and then started again only when the PSA returns to the initial pre-treatment value. This is the result of this trial, which is completed. You can see on the left here that this is the Kaplan-Meier curve. The median time to progression was increased by about 19 months. Overall survival was increased by about 28 months. Importantly, that not only was the clinical outcome better, but the patients did not receive abiraterone about 46% of the time on trial. In other words, only about half of the time were they being treated. The other half, there was no treatment being given. And as a result of this, because abiraterone is quite expensive, there was an average cost reduction of $70,000 per patient per year. There's also an interesting phenomenon that we honestly did not expect, and that is that this long tail here that you can see on the left, we currently still have, I think, 3 patients stably cycling, and they're approaching 10 years in therapy. What I think about that's important about underlying mathematical models to design a trial and to conduct the trial is that those same models can be used to analyze the trial. And I think this is very important. Clinical trials typically now are given, let's just say there's drug A and there's drug B, and the outcome is that drug A is better than drug B with some level of statistical certainty. What is not asked or answered in those trials is why. Why is A better than B? And how could we have done it better? How could we have used either treatment in a more effective way? So the application of the mathematical models to the analysis of the trial, I think, extends our ability to understand that. So just briefly, what we did is used longitudinal data from the trial to update model parameters, and then we applied the model the updated model to every patient in the trial to understand what happened into the underlying populations during the treatment, and I think most importantly, what could we have done differently that could have improved the outcomes. And just briefly, what I'll show you here is that one of the things that we found was we overtreated patients. Our goal was to reduce the population to this PSA to 50% and then stop treatment. But you can see in this graph, what we actually did in this patient was we wanted to stop here, but because of the trial design, we didn't really stop until here. And so you see the PSA was driven to close to 0 in 3 consecutive cycles. And remember now that our goal here is to maintain a significant population of sensitive cells and allow them to suppress the proliferation of the resistant cells. By eliminating the sensitive population, we actually sow the seeds of failure for this individual patient. And so what we've learned from this is that we need to pay more attention to this inflection point. So we then turn attention to castrate sensitive population. This is the initial therapy, and classically this is treated with androgen deprivation therapy, which is effectively is a chemical castration. Essentially, turn off the androgen production by the body, and since the initial cancer cells are typically androgen dependent, you can see a significant and often prolonged response. There has been a recent attempt to improve on this by adding something like abiraterone, which is second-line therapy, to androgen deprivation therapy, and to give those up front as a treatment. The Latitude trial was very instrumental in this change in practice. And what they showed was that if you added abiraterone to the androgen deprivation therapy, that you had a prolonged response. They showed a median time to progression of about 36 months and a median overall survival of 53 months. And I will note for you that our median time to survival in the standard androgen deprivation therapy and then the abiraterone applied with an adaptive therapy was 59 months, already significantly greater than this trial results. So Jinsong Zhang again did a new trial, again pilot trial, looking at the adaptive administration of this combination of androgen deprivation therapy and abiraterone. This was a complex trial, and I won't go into the details beyond saying that the patients, depending on the evolutionary dynamics, could receive no treatment, they could receive one drug treatment, or they could receive both drug treatments, again, based on these the underlying mathematical models. We have gotten to this has accrued the patients that they wanted, so it's close to accrual, and so now we're simply watching them. I guess the good news and bad news is the same news, and that is only 5 of the 16 patients have progressed. So we have not yet even met a median time to progression, but we know that if all of them progressed immediately today, that the median time to progression would be 46 months, already substantially longer than the Latitude trial. We know that patients receive no therapy more than half the time. There are no drugs at all, and they receive both drugs only about 14% of the time. So again, since abiraterone is an expensive drug, this significantly reduces the financial toxicity, so-called. And so with that, I will thank you. I will give a plug to the Moffitt Evolutionary Tumor Board, in which we actually have mathematicians and evolutionary biologists actively participating in clinical decisions about individual patients. And with that, I'm happy to answer questions.